Hello, everyone, and welcome to today's webinar entitled Applications of Humanized Mouse Models in Preclinical Research. My name is Jian Hong. I'm a Senior Marketing Manager here at GenoBioTX. Before we begin, I'd like to invite everyone to keep any questions to the end of this session or write them in the chat box. We will address them in the end of the webinar. Today, the webinar will be presented by Dr. Derek Resnick. Dr. Resnick earned his bachelor's science degree from Barrett, the honor college at Arizona State University, and completed his PhD in neuroscience from Baylor College of Medicine. Throughout his research career, Dr. Resnick has focused on the preclinical implications of developing and identifying translational biomarkers for mouse and rat models of diseases, including schizophrenia, Parkinson's, and Alzheimer's, and rare genomic imprinting neurobehavior disorders. He's an expert at the preclinical characterization of novel rodent models and the discovery and isolation of robust, reproducible biomarkers for evaluating preclinical therapeutic interventions. Early in 2024, he started his current position as a business development manager within the industrial clients department here at GenoBioTX. During this webinar, Dr. Resnick will discuss the impacts and breakthroughs of humanized mouse models in preclinical drug development, highlighting key strategies and examples in this field. With that, I will turn it over to Dr. Resnick.
Thank you for the great introduction, Jane. Let me share my screen real quick. Okay, so everyone can see my screen?
Yes.
Okay, great. Okay, so as Jane said, my name is Derek Resnick. I'm a business development manager here at GenoBioTX. Thank you for everyone for joining the webinar today. We're going to learn about some of the applications of humanized mouse models, especially in preclinical drug research. So here's just some overview of today's webinar. So I'm going to start off with a brief introduction into the general overview of what is a humanized mouse model. Then I'm going to give some examples of humanized models that we have done here ourselves at GenoBioTX. And then we're going to learn, and I'll show some more examples of how to generate a human-like immune system. So it's another version of a humanized mouse model. And then I'll finish with a summary. So to start, some background on what makes a humanized mouse model. So unfortunately, a humanized mouse model is not a mouse wearing a cute little bow tie. In reality, a way to generate a humanized mouse model can be several different ways, but the first way is through genetic engineering, by which a piece of human DNA can be inserted into the mouse genome. And so this can be either full sequence of a human gene or parts of a human gene. Another way to make a humanized mouse is by reconstructing a human-like immune system. So by engrafting and injecting human cells into an immune deficient mouse, we can generate something close that resembles the human immune system in a more controlled environment like the mouse species. Another way to generate a humanized mouse is by engrafting human cancer cells into mice. And so this allows us to study the dynamics of human patient cancer cells, as well as doing preclinical therapeutic screening. And lastly, is we can reconstitute the human microbiome in mice. So these are mice, again, that are immunodeficient, but also they do not have a microbiome anymore in their gut. So by taking patient microbiome samples and inserting them into these mice, we can get a better idea of how the human microbiome operates, again, in a more controlled laboratory environment, such as the mouse species. And so why would we want to use humanized mice when we already have thousands of genetically engineered mouse models that are widely available to us? One of the really good advantages is that we can compare different tumor therapies. And so we can actually take human patient biopsy samples from their tumors and then engraft them into many different mice, which then allows those tumors to grow, and we can do a wide variety of preclinical therapeutic screening. So then when we can identify drugs and compounds that act on these tumors and are able to reduce them or eliminate them in these mice, then hopefully we can then translate them back into the clinic and into these patients. Another way to use humanized mouse models is in order to study the human immune system. But we do this by, and I'll touch more on this later in the webinar, but very briefly, we can inject immune deficient mice, so mice that don't have immune cells. We can inject them with two different types of human cell lines or cells from human patients, which then are able to propagate inside the mouse organism, and then it essentially constructs a human-like immune system, which then can be later used to study the immune system, or again, more preclinical therapeutic testing. And this is more for the genetic side of humanized mice, is that we can help better understand the basic biology of human genes, but also what happens when these genes are implicated in different forms of disease. And so you can see here, this is just a small list. It's not expansive to all the options that we have, but we have many different mouse models and research domains that can be studied at GenoBioTX. So this is things like immune oncology, different types of autoimmune diseases, diabetes models, as well as Alzheimer's disease, and even COVID-19 humanized mice. And so feel free to take a picture of this QR code or go to our website. There'll be more information at the end, but we have an expansive list of humanized mice. And moving on into some different examples of the humanized mouse models that we've created ourselves here at GenoBioTX. And before I start, I do just want to mention that we do have more than 600 strains of humanized models available, and this list keeps growing. Here are just some examples. We do have both mouse and rat humanized models, but for today's webinar, I'm just going to focus on some examples from our mouse models. But we do have systems that have a single construct, a single humanized construct inserted into the mice, but we also have models that have 2, 3, and sometimes even 4 humanized genes in the mouse genome. So it really just depends what kind of research model that you are looking for. But to get started on some of our examples, I'm going to focus on a really big hotspot in the field of just science and also humanized model systems is humanizing different immune checkpoints. And immune checkpoints, very briefly, are protein interactions between T cells and different cells in our human body. And so essentially, when a T cell interacts with either a antigen presenting cell or something like a tumor cell, a protein on the T cell's membrane is able to bind and interact with another protein on these other cells. And this interaction can either be co-inhibitory, so it can either tell the T cell to deactivate and to not destroy the cell that it's interacting with, which is a good thing because you don't want our immune system going around destroying everything that it comes in contact with, but also they can be co-stimulatory, which tells the T cell to become activated and to tell the immune system to destroy the cell that it's interacting with. And again, this can be a good thing because when cells are undergoing stress or if they're old and they need to be replaced, this is a good thing for the immune system for just cell turnover. But also many of these immune checkpoints can also be dysregulated, especially in disease types such as cancer, which can lead to either immune evasion or proliferation of the cancer cells. And so at GenoBioTX, we have many different immune checkpoint mouse models. This is not an expansive list, but we do have many available, several hundred, and we are also actively developing more. So always check back every couple of months that we are going to be adding to our list of our humanized mice. And so for our first example, I want to look at PD-1. So this immune checkpoint is also called, like its full name is programmed cell death protein 1, PDCD1 or PD1. So its natural function is that it binds its ligand, PDL1. And so PD1 is actually on the T cell, and PDL1 is on frequently an immune cell that it's interacting with. And so this is actually a co-inhibitory signal. So this is something that prevents T cells from destroying normal healthy cells. And so this is a normal, healthy, everyday process that's happening all the time in our bodies, and it prevents T cells from killing healthy cells. However, a problem is that cancer cells can upregulate this PDL1 in order to evade T cell based killing. So this is an issue in cancer. So they'll make more of these PDL1 ligands, which prevents T cells from activating the immune system to destroy tumor cells. A solution to this is that we can inhibit this binding between PD1 and PDL1 through targeted antibodies. And so this allows the T cell to not have this co-inhibitory signal, which then allows it to become activated and then leads to the cell death of the tumor cell. And so we've actually generated a humanized PD1 mouse model. We did this through CRISPR-Cas9 technology, where you can see we generated a cut site at the beginning of the mouse PDCD1 gene, and then we, through homologous recombination, we inserted the human PDCD1 cDNA sequence into the beginning of the mouse gene, so creating a humanized knock-in allele. And here's some validation data from this mouse line, and this is FACS data from mouse spleen lymphocytes, and you can see here that in this unstained control, when we're looking at human PD1 and mouse PD1 proteins, this is unstained, so we're not able to detect anything. But in the wild type animal samples, we can see that we can only detect the mouse PD1 because the human PD1 construct is not there, so that makes sense. But when we look at the homozygous PD1 knock-in animals, we can only detect the human PD1 protein and not the mouse PD1 protein, demonstrating that we have created a successful humanized PD1 mouse model. And then we can further use this mouse model to do efficacy study testing. And so we took these homozygous animals and we engrafted them with tumor cells. So from an MC38 line, this is a colon tumor cell line. And after those animals have tumors engrafted, we can then try to treat their tumors with a anti-PD1 antibody. So we used Keytruda. Keytruda is a commercially available human anti-PD1 antibody on the market for cancer patients. And you can see at 3 different dosages, either at 1, 3, or 10 milligram per kilogram, you can see that the higher the dosage, the more effective the anti-PD1 antibody is at preventing the tumor growth. And we also did this in a second anti-PD1 antibody, by which we used Opdivo, again at a increasing dosage of where you can see the highest dosage of 5 milligram per kilogram, again has the highest efficacy of reducing the growth of the tumor cells. So it demonstrates that this humanized mouse model is able to be used to study different cancer therapeutic drugs. And it's also important to note that both of those studies were done in C57 black 6 animals, but we also have our humanized lines on different strain backgrounds. So this is just another study, but this time the strain background for this humanized allele is on a bulbsy nude mouse. And so just a different type of strain that is used in preclinical settings. But you can see again that when we use Keytruda or an entinostat, and also a polytherapy of using both the antibody and the entinostat, you can see that the polytherapy is the most robust and successful at reducing the amount of tumor growth in these animals. Now moving on to our next immune checkpoint molecule. So we're going to look at CD47. So CD47's natural function is on healthy cells. So it's a protein that's on healthy cells, and it interacts with its ligand, the SRP alpha protein, which is on macrophages. And this interaction acts as a stop signal to prevent macrophages from killing healthy cells. So again, this is a good function for this immune checkpoint protein. And this binding essentially tells the macrophage, please don't eat me, do not phagocytose me because I am a healthy cell. However, the problem is, again, cancer cells can upregulate this inhibitory signal on their cell surface, which actually tells the macrophages, don't eat me, even though I'm expressing a warning signal. It gets overridden in this balance, so it ends up not being phagocytosed. A solution to this, again, we can use a targeted antibody against CD47, by which it prevents this CD47 and SERPA alpha interaction from occurring, which leads to only the eat me signal from being picked up by the macrophage. So this swings the phagocytosis signal into the eat me side, which then allows the macrophage to engulf this tumor cell. And when we generated our humanized CD47 mouse model, so we actually inserted the second exon for the human CD47 gene into the mouse allele through homologous recombination. And we did the second exon because this encodes just the extracellular domain of CD47. So this would be the part of the protein that is on the outside of the cell surface, which the antibody or a potential drug could interact with. And some validation data from this animal model. You can see here, this is all wild type cells from either lung, brain, or spleen. And you can see on the x-axis is the mouse CD47, and on the y is the human CD47. And you can see in the wild type animals, we can only detect the mouse form of the protein, which makes sense because the construct is not there. But when we look at the homozygous knock-in animals for the human CD47 allele, we can see on the x-axis is now the human CD47 target. We can only detect the human form of the protein and not the mouse. So again, showing that we have successfully generated a humanized model. And when we look at an efficacy study for this line, again, these animals are engrafted with colon tumor MC38 cell line, but this time we use an anti-CD47 antibody, and this one is a humanized antibody from 47 Incorporated. And you can see at two different dosaging levels, one at 10 mg per kg and 20 mg per kg, you can see that they do significantly reduce the tumor's capability of growing. However, when we do a polytherapy with our CD47 antibody, as well as a PD-1 antibody, so something that we just saw in the previous model, we can see that it further significantly reduces the amount of tumor growth in this model, suggesting that maybe you can get a even better response by blocking CD47, but also doing a polytherapeutic approach, which is often what happens in cancer patients. And so our next model that I want us to look at is the human NT5E gene. So this is the 5' nucleosidase, also known as CD73. And so its natural function is to act as a ectonucleosidase, so a nucleosidase that's on the outside of the cell membrane, and it converts AMP to adenosine. And again, this is a normal, healthy process that happens in our body all the time. Because when we have extracellular ATP, it gets broken down and further broken down into adenosine, which can then be recycled for the cellular environment. However, a problem is that when cells die, they can rupture and release large amounts of ATP into the extracellular environment, which can end up leading to large extra amounts of adenosine, which actually naturally acts as a immunosuppressive agent because it'll bind to immune cells and tell them to not become active. And this also is a natural and a good process, because if you have cells that are dying naturally and are releasing this ATP, you don't want your immune system to continue this process. You only want to go after the cells that are supposed to be being destroyed. However, in cancer, this could be a further problem because the tumor cells that are being destroyed are going to be releasing ATP, but it's also going to be downstream telling the immune cells to become immunosuppressed, which then it prevents more tumor cells from being destroyed. So in order to keep that cytotoxicity occurring, one approach again would be to try to inhibit the conversion of AMP into adenosine by blocking the function of CD73. And so this is just the schematic diagram of our humanized CD73 or NT5E mouse model, by which we knocked in the human cDNA of this gene into the mouse allele. And you can see here more validation data. Again, so this is FACS data from wild type mice. And you can see in both CD3, so total T cells, and also CD4, which is just helper T cell populations. When we look at MD or mouse CD73 protein levels, we can clearly detect them because it makes sense because it's wild type animals. And when we look in the homozygous humanized CD73 knock-in mice, we are unable to detect the mouse CD73, which is good. And then vice versa, when we look at the human CD73 in the wild type, we cannot detect it in both the CD3 and CD4 cell populations, but we're able to strongly detect the human CD3 protein in, again, those total T cell and helper T cell populations. Also, we can detect them very strongly in the cytotoxic and inhibitory T cell population through a CD8 marker. So again, successfully confirming that we've made a successful humanized mouse model for this gene. And then when we do an efficacy study with these animals, again, we do tumor engraftment. This time we used several different CD73 antibodies, and you can see 3 different CD73 antibodies at the same dosage level, all have a very comparable reduction in the tumor growth of these animals. And we also have many more humanized mouse models, not just immune checkpoints. We have many other targets, including cytokines, which I want to focus on right now for my final example. So I want to look at the cytokine interleukin 23. So again, this is a normal functioning protein that promotes an inflammatory response in several different organs. And this can be for a normal, healthy reason, but it can also go poorly and become exacerbated in different conditions like plaque psoriasis and Crohn's disease, among others, depending on the organ that it's being that it's interacting in. And so in plaque psoriasis, when you have a stimulus on the skin that leads to the activation of either macrophages or dendritic cells, which lead to the expression of IL-23, so this interleukin, this cytokine, which is the top of the cascade for more immune cells that release more cytokines, which actively activate a different type of skin cell, which then releases its own host of pro-inflammatory cytokines, which then generates skin remodeling, which leads to the psoriatic plaque that forms on the top of skin in plaque psoriasis. And so essentially this just becomes a vicious cycle that one of the solutions to this condition would be to block IL-23, again, through targeted antibody approaches. And there's already some on the market, many of which that we all have probably heard of on TV, such as Tremfya or Stelara. But here's a diagram of the humanized IL-23 mouse model that we've generated. So again, so this one was actually through embryonic stem cell based gene targeting, by which we knocked in and replaced the mouse allele with all of the human exons, as well as we used a neomycin selection cassette, by which we were able to select the positive cells after we did our homologous recombination, and we were able to remove the selection cassette because it was flanked by FRT sites. So when exposing it to a flipase, we're only left with the human target allele. And when we look at the validation data, we screen for human IL-23 levels in the serum from these animals. We can see that we are unable to detect the human IL-23 in the wild type animals, but we can strongly detect the human IL-23 in the homozygous knock-ins. And vice versa, we cannot detect the mouse IL-23 in the knock-ins, only in the wild-type animals, again, suggesting that this is a successful humanized mouse model. And the way that we can validate this model is actually through a psoriasis-like condition in mice, and by which we expose animals to imiquad or IMQ, which induces a psoriasis-like phenotype. So you can see here in our control group, the top layer of hair is gently removed from the animals, and Vaseline is exposed to them, which does nothing. But in our test group, you can see that when they are exposed to IMQ, they begin to generate a psoriasis-like phenotype on the top layer of their skin, similar to what we see in humans. And then when we treat them with an IL-23 antibody, you can see the significant visual improvement in the psoriasis phenotype. And this is quantified here through a PA-SI area score, by which you can see in green is this group 2, and has a severe area score difference between the treatment group with the anti-IL-23 antibody. And again, just for basic understanding, anything above a 5 is generally understood to be a disease relevant score. So these animals are having significant improvement from the anti-IL-23 inhibitor. And we can also see this improvement at level of histology. So we can see our control group has a very thin epidermis, whereas when we see the psoriasis-induced group, you can see it is significantly thicker and inflamed. And you can see this in the pathological score, the increase, whereas when we treat them with the anti-IL-23 inhibitor, we can see that the epidermal thickness and the pathology score both start to reduce to more manageable levels. Okay, so switching gears, so another way that we can generate a humanized mouse model is by reconstructing a human-like immune system. And so just a quick little background, we don't have the time in this webinar to discuss the entire history of immune deficient mice, but essentially in the 1980s, we had 3 primary mouse models: a skid mouse, a nod mouse, and a nude mouse. But since then, we've generated tens, if not potentially hundreds of different iterations of immune deficient mice, which essentially have different levels of cell depletions of different immune cells. But at GenoBioTX, we have our own in-house generated immune deficient animal. We call it the NMG mouse. So it's a NOD-CG background with the SCID mutation in the PRKDC gene, as well as the mutation in the IL-2RG gene. And this confers a high degree of immunodeficiency in these animals, but also allowing them to have a long, healthy lifespan. And these mice are a great tool in order to do either cell or patient-based xenograft modeling, as well as they are very well equipped to reconstitute a human-like immune system. But I also just want to quickly share that these animals have been validated with more than 85 different human cancer lines. And here's just some quick validation data on these animals before we use them going forward. You can see this is FACS data from the spleens of these animals. So you can see when compared to traditional immune deficient models like the bulb-C nude mice or NOD-SCID mice, you can see our NMG mice have a severe depletion in the B cell, the T cell, and the natural killer cell cell populations. And this is also can be seen here graphically, where the NMG mice don't even register on the scale. And this is also seen in a very similar pattern of when we look at the peripheral blood in these NMG animals. We can still see these 3 cell populations are severely depleted. And again, as I mentioned, these animals have long, healthy lifespans. They have normal growth, so they are a very robust system to be used, especially for reconstituting the immune system or doing different types of CDX and PDX modeling. And so As I mentioned, in order to generate a human-like immune system, there's two primary approaches. The first approach is by taking an immune deficient animal, such as our NMG mouse, and injecting it with human peripheral blood mononuclear cells. And then you can wait about 3 to 4 weeks. And then these cells will have generated and repopulated the immune deficient animal. So you've effectively generated a human-like immune system in a much more controlled environment, like the mouse species, by which you can then study different types of infectious disease, do different type of gene therapy studies if you want, or do different drug target studies. Some pros of this model is that it's easy to establish. It only takes about 3 to 4 weeks, so it's very short time in order to start doing your studies. You get very efficient T cell engraftment. However, some trade-offs is that your myeloid cell engraftment is poor, whereas if you use this system chronically in the animals, you will eventually have a lethal graft versus host disease set in. So these animals will eventually die if you use it too long. So these animals are more generally used for shorter time point studies. And then the second approach is using human hematopoietic stem cells. And so you can take younger animals and you can irradiate them as well. And then you follow that up with injecting your HSC or your CD34 positive cells. How it does take a little bit longer to allow this system to develop into a human-like immune system, so about 3 to 4 months. But once you get these animals, you can do very similar types of studies. Some of the pros is that this model actually develops multiple hematopoietic lineages from the use of the HSCs. You can elicit a primary immune response in these animals. However, the T cells that have been generated are restricted to interacting with the mouse MHC. But so starting with an example of the human PBMC modeling, one thing you can do in these animals is you can screen donor cells, so you can determine which human patient PBMCs end up being the most robust and have the highest percentage of in the mouse models, and you can do this over the course of about 4 weeks. So at the same time point, you can be measuring their body weight to try to determine which donor is going to be the best for your studies going forward. So based off of this data, it would appear that based off of the percent of human CD45 cells, as well as the most standard body weights, it appears to be donor 5 would probably be the donor PBMCs that you'd want to use going forward. And also you can evaluate all the different cell populations from your broad CD45 cells, so your PBMCs, as well as your total T cell populations, your helper T cell populations, and your cytotoxic T cell populations. And again, this is all from FACS data. But then you can do further preclinical testing in these animals by after your PBMC engraftment, you can then engraft them with a tumor. So something like, as we saw earlier in the webinar, in this case, it was a melanoma cell line, and then you can do your initial testing dosage followed by your body weight measurements. You can see here when testing 3 different antibodies in this myeloma cell line after being engrafted with the PBMCs, you can see that has a significant reduction in the amount of tumor volume in these animals, again, with pretty standard non-fluctuating body weights again. So showing that this model does work for this kind of experimental validation. And then when we look at the hematopoietic stem cells, so our CD34 reconstruction model. So again, this is the one that takes a little bit longer, but these animals can also be tested out much further. As you can see here, again, in FACS data, you can detect the total CD45, 3, 4, and 8 cell populations. And also, I just want to mention, you can take these animals out much further since they are less likely to have a graft versus host disease set in. So these animals can actually be measured out closer to 6 months compared to maybe a month or two for the previous system. And again, you can do very similar, but also more longer time point studies when you can engraft them with tumor cells, do your therapeutic testing, such as an antibody, potentially see the improvement over a longer range of time, as well as you can do your safety and toxicity measurements as well. And also a benefit of this model is that you can actually generate a pretty robust natural killer cell response. If you inoculate these animals after you've generated your human-like immune system, if you inoculate them with human interleukin 15, so this is an activating cytokine for natural killer cells. You can see here that in red, so this is the HIL-15 mice, you can see the significant increase in the proportion of the activated NK cells. And then we can also see this by FACS sorting data. So this is the total human CD45 cell population in these activated animals. And you can see that we can detect through the CD16, CD56 cell markers, the significant increase in the activated NK cells. And with that, so we've learned very briefly today that a humanized mouse is more of a broad term that can refer to several different types of models. It can be a mouse that has a few human cells in it. It can be a mouse that has a strand of human DNA incorporated into its genome, so a genetically engineered animal, or it can be a model that has a human tumor engrafted into it, or a mouse that has a humanized immune system. So it's several different, and there's also many other different types of models that we haven't touched on in this webinar. But we also reviewed some examples of the genetically engineered models that we have in-house at GenoBioTX, and these were some immune checkpoint targets, as well as a few cytokine and other targets. And then lastly, we learned and reviewed about how to reconstitute the human immune system, or a human-like immune system in mice, and doing that through two primary approaches, using either PBMCs or HSCs, so two different human cells. And so in summary, at GenoBioTX, we aim to be your professional, comprehensive, and easy to use partner to provide you with genetically engineered animal models and also preclinical CRO services in order to best support all of your research needs. We also aim to be a one-stop service provider. So whether that's you would like us to generate a custom genetically engineered animal model, we can do mice, rats, cell lines, zebrafish as well, or we can provide you one of our 1,000, we actually have 10,000 off-the-shelf mouse lines available, and those are genetically engineered and also immunodeficient animals, similar to the ones that I presented here today. But we can also provide on the CRO side breeding services and phenotypic analyses. So we can do rapid colony development, as well as we can do many different types of CRO preclinical experiments, including but not limited to CDX and PDX modeling. And with that, thank you for attending today's webinar. And if there's any questions, I'll be happy to answer.
There are no questions in the chat box.
Yeah, I think we are done for the webinar. The online audience left already.
Oh, okay.
Yeah, so all from our company. It's okay, very good presentation. I think if we put it on YouTube, maybe we can get a lot of fans for this presentation, I'm sure.